Still scheduled for Wednesday next week, right? Boy, at first, this section, both of my classes, this one and AP1 seem to just, the second section just flies. So let's see if we can get started on the, or continue on what we were talking about, the cell. Finish up, hopefully we'll be able to finish up the cell so that we have at least a full 2 days to talk about membrane and membrane function. All right? So, after today, there's probably going to be a quiz open online on the cell. All right, it's not open yet, but it will be. Okay, so, the cell. We left off right at the cytoskeleton, am I correct?
Yes, you are correct.
All right, quick quiz. Yes. Mastering biology. There should be, and I believe it's open until the next Tuesday. I think I just said, I'm to heck with it. I'm just going to leave it open until the night before the test. Just don't wait till that point. Yeah, I just thought it would be easier, but you got to do your part and actually start on it sooner. Otherwise, it gets really confusing. I hate you having to do homework right before test. All right, quick quiz. And there's a couple answers to this, and I want to see a hand raised. Give me one similarity between chloroplasts and mitochondria.
They both have their own DNA.
They both have their own DNA. That is correct.
Isn't the process they perform just reverse of each other?
Almost.
Almost.
Not necessarily. But yeah, and what process would you, if you put that into a grand scheme, you would call their function
Energy synthesis or energy
transformation.
transformation.
Yeah. Ooh. All right. What's that? You got a new one?
Don't they reproduce themselves? Yes.
True. Hold that thought. I just want to cover this one before we move on. All right. Some people think they're the opposite, where like a mitochondria takes the result of carbs and produces ATP. Okay. A chloroplast takes light and CO2 and makes carbs. Okay, it looks backwards. However, plants also have their own mitochondria. Okay, they have their own mitochondria. So they then take their own carbs and metabolize them just like this to make ATP.
So one's just earlier on in the...
Right. This is not the opposite of that, and it's also not, this is what happens in animals and this is what happens in plants. It's photosynthesis and the chloroplasts are to plants what eating is to animals. Both eating and photosynthesis gives the host fuel to then metabolize. We'll cover that in the next unit, but all right, but basically it's energy transformation. All right, and you said, hello? And you said, we moved on, you said,
They reproduce themselves.
they reproduce themselves. Exactly. That kind of goes along with having their own DNA. Okay? So they reproduce themselves, they have their own DNA, they're both involved in energy transformation. Anything else? There's an obvious one.
They're double membrane.
There you go. They both have at least 2 membranes. Mitochondria have 2, chloroplasts have 3. Right? They have outer, inner, stroma, thylakoid membrane. And there's one left. A bit more complex. Not as clear. How about their origins?
They make both prokaryotic cells.
Yep. It's believed that both were originally prokaryotic cells that have entered into a symbiotic relationship that they can no longer leave. They've now become basically stuck there. But plants are way better off having chloroplasts, and animals are way better off having mitochondria. So everyone wins in that relationship. All right. So, so much, that was what we covered last, that and peroxisomes. But anyway, the cytoskeleton is one of 3 areas that probably has changed the most since I was in your seat. Okay, the 3 areas are the cytoskeleton, genetics or gene expression, and cell communication. Those 3 areas are probably the 3 cutting edge areas of cytology or study of cells. Now, when I was in your seat, we knew these existed, but we didn't know to what extent, and we didn't really know what their purpose was. Some of the pictures that are in this book were cutting edge when I was when I was a student. So I'm just saying, there are pictures, and I remember just like kind of gee whiz, like wow, that is really a cool picture. What does it mean? We now know what it means. Now, the 3 types of fibers are microtubules, microfilaments, and intermediate filaments. Now, these are general terms. We will have specific terms that we will use for what they make, okay? But if they were wood, we'd say like one is a 2x4, one is a 4x4, and one is a 2x6, right? They are used to make other things. And once we make other things with them, then they take on a different name, right? It might be a pillar, might be a roof, might be a scaffolding, might be a whatever. Okay, so these are the basic names, and then we're going to come back and show what we actually make from them. They will help define the cell shape. They're also involved in a lot of types of movement, either of the cell, of the external part of the cell, stuff around the cell, or within the cell. Okay? So there's a big combination. Now, the one thing I want to remind you is that they don't themselves move things, but what they do is they act as like a train track. And I'll describe that a little bit better coming up here. Now, first of all, a microtubule. These are the largest of the cytoskeletal aspects. And it looks like a giant hollow tube. Now again, you got to remember we've blown this up significantly. But this tube is made of a series of repeating proteins called tubulin. So if you want to make the tube longer, you simply add more tubulin molecules. You want to make it shorter, you simply take away. And it's simply just a spiral. And to make it shorter, you simply spiral back. All right, so they're not impossible. They're not really, they're not carved in stone. So these things can grow and shorten relatively easy. Not as easily as other fibers like the microfilaments, but they still can happen. All right. Now, keep that in mind. It's basically a tube, and you can see the end of it right here. Now, one of the things that they can do is they are associated with what are called dinins or motor proteins. And these dinins can walk along a microtubule. Now, it just matters of what those molecules are attached to. If they are attached to an organelle, they might be able to carry that organelle around using the microtubule as sort of a track. On the other hand, if they are attached to another microtubule, this happens. Like that, where they're moving against each other. Now, I'm sure you're looking at that going, unless you want a really big slide rule, what good is that? What would be the effect of this? Well, this, not much. However, because you need to link them together. If they are linked together, do this. Now, move your left finger up, your right finger down, and back, and vice versa. What happens? Friction, but don't your fingers start going like this? I'm exaggerating a smidge, but don't they start doing this? That's what happens. Now can you see what's going to happen here? If they start moving backwards and forwards, what are they doing? What is it? It's a tail. All right, so when you get it moving back and forth like that, you can get these things to bend. If you've ever seen a tail on a cat that have really long tails, they're kind of weird because you look at the very end of the tail and the end of the tail goes, and like this, right? Or like a gibbon, they can grab things with their tail and it'll wrap around a branch or something like that. Same sort of idea, a little less flexibility, but nonetheless, they're able to wave back and forth. And that's how things swim with these structures. All right, so once we know what a microtubule is, we can then look at how they are used. And they are used as a substructure for larger pieces. Sometimes there's short segments, other times they're very long. All right, we're going to start with with the spindle apparatus. These have the least amount of description, but these are part of how cells divide, and they form a cage, kind of like this, around the DNA. And as these things move apart, they help to divide the molecules of DNA in half. One set pulls one way this way, one set pulls the other half this way, and it helps to divide the DNA. We will cover that way more detail in Unit 4 when we look at mitosis and meiosis. Now, as far as centrosomes and centrioles are concerned, this is something I will warn you is usually very often mistaken. Now, what are they? They are microtubule organizing regions. So remember when you make this cage, there's going to be one here, and there's going to be one here, organizing them. And they are in themselves made of microtubules. If you notice, there is a series of triplets. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 trios of microtubule. And each one, and these are found in animal cells, you have a pair of them. Okay? And there are 9 trios. And if you notice, centrosomes are composed of a pair of centrioles. So each of those cylinders is a centriole. And in fact, when they during normal behavior, they will divide and a new one is made for each half. So anyway, the point is, each of these pieces is made of 27 microtubule segments. Something else I want to point out is that they also look like what is called the basal body of flagella. So when I talk about a flagellum, I'm going to point to something and say, same structure. All right? Now, moving on to the next one, I said there was another usage. Flagella and cilia. The only thing that's really different is slightly different mechanical structure and slightly different numbers and slightly different sizes. These are actually visible. Now you have to have a good microscope to see these things. But how many have labs so far? A couple of you? Were you able to see the paramecium? You couldn't see the cilia though, could you? No, they're moving too fast. Imagine, well, you know the crew boats, like that have the big oars? Imagine if they were traveling about 100 miles an hour. How fast the oars would have to be moving? Do you think you'd be able to see the oars? Not really. Now make them microscopic. That's kind of how these work. The ciliates are cells that use cilia to paddle themselves around. Okay? So in this case, they simply wave their cilia and can actually swim. And those of you who saw them under the microscope, they are good. They are good swimmers. Other ciliates might look like some look like a pitcher, and they have cilia around the top, pulling food in. The cells of our trachea are lined with ciliated cells. I should say our trachea is lined with ciliated cells. Now, pretend we're at a concert. Everyone put your hands up in the air. You know, and wave them around like you just don't care. No, I'm just kidding. No, put them up. Now, follow me. Well, you know, this is very... Now, if you work at the same time, people are walking by and going, what are they doing? Now, imagine if there's something up here. What would happen to this material? It would get moved. So let's say there's a big glob of mucus hanging over everyone's head. Wouldn't that be disgusting? You get this big glob of mucus, and all of a sudden we start going like this. What's going to happen? That mucus is going to be moved. That's what happens in our trachea. We line the trachea with mucus. That mucus traps dust dirt, toxins, things like that. And what do the cilia do? Since the cells don't move, the cilia moves the stuff. Pushes it up into the back of our throat. We either cough it up, we swallow it, digest it, and it's gone. It's protective. In the female reproductive tract, there are stretches that are ciliated. Since egg cells do not have a mechanism for movement, they create a current. And the current draws the egg into the fallopian tube and moves it along. Okay? Those are cilia. They're shorter and more numerous. There's a slight structural difference, but flagellum, on the other hand, are quite large. Okay? They are long and fewer in number. In fact, in humans, that's the only cell that has a flagellum. And what is that? That's a sperm cell, correct. We call it the tail. And how does it move? Like I said, just like this. So a eukaryotic flagellum moves back and forth, and it acts like an engine on a boat. That's all it does. Now, if you saw a flagellum in a bacterial cell, they move like this. They're definitely different structures. Okay? So, what have we had so far? Microtubules help to make the spindle apparatus. They make centrioles and centrosomes. They make flagella. They make cilia. Okay? And this is the difference between cilia and flagella. Numerous, that's the fact. There's fewer flagella, more cilia. But look at the difference. Here, you've got a sequence of pairs, and if you notice, the pairs are connected by motor proteins. And you can kind of see it up here. So as they move back and forth against each other, you get the flagellum and the cilia waving like that. All right? Now, the difference between cilia and flagellum is cilia do not have this pair in the middle, flagellum do. So you got 9 pairs around 2 or 9 pairs around 0. All right? And if you look real close, you notice that they have an anchor. And that anchor is right there. And doesn't that anchor look familiar? That's the basal body. That's the structure that is almost identical to the centrioles. Now that's not surprising. In fact, that's an interesting piece of evidence in support of evolution. What it shows is that not every article has the same function, even though it has the same structure. There's a great argument. Somebody said, well, how do you take a piece away from one structure and make it functional? Like a spring-loaded mousetrap, right? You can take one piece away and that mousetrap's not going to work, right? What if we take the little stick that holds the spring down? Would that function? Oh yeah, it is. I saw it in a trial. The guy goes, it's perfectly functional. He opens it up, sticks it on his tie, and says it's a perfectly functional tie clip. So it doesn't mean it has to have the same function, it just is useful for something else. So there's a basal body, looks just like a centriole. Could come from the same genes. Possibly. All right, so far so good. And again, just looking at this, we have the motor proteins or dinins, and as they pull against each other, as long as they're, see the connection right here? As long as they're connected and they can't just simply slide by each other, they'll do this. And once they start doing that, we start getting this. And once we start getting organized movement, now we can use them for whatever purpose we need. But one more thing you got to look at. Every time one of those proteins moves, just once, it requires ATP. And there's about 18 of them on there right now. And if you had hundreds of cilia moving constantly, it's quite expensive. So that's a big use of ATP if they have them. All right? Now, that's microtubules. Remember, microtubules were the individual little pieces for building cilia and flagellum and centrosomes and centrioles and the basal body and the spindle apparatus for cell division. Now, microfilaments, similar in structure but much smaller. That's why we call them microfilaments. They're not microtubules. It's not a tube. it's simply a solid fiber, and obviously it's not as wide. Okay? Now, if you know anything about muscle, muscle is an interaction between two types of fibers. One is called myosin and the other one is called actin. And what happens is they move against each other like this. And if you notice, the two ends are getting close together. That's how muscle works. These whole long lines of these fibers all overlap each other and the muscle shortens. Muscle shortens, you pull on a bone, bone moves. So, actin filaments are an example of a microfilament. All right, they're not all the same, but they're an example. And now, what do microfilaments do? One of the things they do is they have the ability to change in size. So what they can do is, let's say you are a, did anyone see amoeba in lab? You saw, did you get a good picture or not? No? Did you see it?
Yeah.
Okay. Sometimes you get good views, sometimes you don't. It's kind of a hit or miss thing. But my example I want to give you is imagine a train, and the train doesn't necessarily follow solid tracks, but in fact can put their tracks down wherever they want. You know? So they put the two tracks here, they drive over it, they pick the tracks up, and then they move them over here, and they put them down here, and they go there, and they keep doing this, they can go wherever they want, right? So what an amoeba does is they build microfilaments, and they simply keep adding to the end of that microfilament. And as that happens, it will push the end of the cytoplasm away. Well, if this is an amoeba, all of a sudden you'll start to see this. And it'll keep getting longer and longer, and then this side keeps getting shorter. Like this. And then maybe it'll start growing another process in that direction. And this one will go over here. And they keep spreading out. And as one moves this way, the rest of it keeps getting smaller. So they ooze. And in fact, it's so cool because you can actually see parts inside moving around. And if they have some food in there, you'll see that moving around. I saw one time it had eaten another, a smaller ciliate. So it was stuck in a little vacuole inside the cell as it was moving around. It was pretty neat. But anyway, so it allows for pseudopod formation. What does that mean? It means false foot. So it looks like when these things push out, it looks like little legs, but they're not. It is the functional unit of muscle contraction. It is the core of microvilli. Remember, microvilli are those finger-like projections on the surface of a cell. So the edge of the cell looks like this. These would be microfilaments. So the core of them would be constructed out of them. Another thing that happens when cells divide, you have two nuclei, and they're going to divide right here. So what you will see is all of a sudden the cell will begin to pinch in, like that. Really what's happening is it's a ring of these filaments that are stuck on the cell membrane, and as they get shorter, what do? They simply pull the cell membrane in and keep pulling it in and keep pulling it in and keep pulling it in until they literally snip the two cells in half. That's how cells divide. It would be like if you took a balloon and squeezed it in the middle, and when you pinched it all away, it would separate. That's what microfilaments can do. Also, it's all the last thing involved in cytoplasmic streaming. If you're lucky, you can see this in some plant cells under the microscope, where the cytoplasm, you can actually see the chloroplast moving inside the cell. When I was in your seat, we didn't know what was causing that yet, because we didn't see that this cytoskeleton was so extensive. Okay, so microfilaments, quite varied in their function. Okay, and finally, intermediate filaments. Now, you notice the very first line says a diverse group, which means showing you a picture wouldn't really be so good because it wouldn't be a good example. But the point is, they're a little bit bigger than microfilaments, they're smaller than microtubules. And they have many different functions. But the one thing they probably all share is they are, first of all, more permanent than either of the other two. And they're pretty much all about stability. I think of it like a cable or a rope, all right? Now, one way that these work is, if these were the microvilli, if these are microfilaments, intermediate filaments would be down here, stabilizing them. Or, let's say this. This is a desmosome. I'm going to come back and talk about a desmosome in just a little bit. But a desmosome is a big wad of protein stuck to the inside surface of a cell. And it would be attached to another desmosome on the next cell over, like a rivet. All right. Now, this right here is another desmosome attached to a cell in that direction. But internally, that desmosome could have intermediate filaments running from it to here. What would it do? What would be the purpose? Think of a tent. Think of carnival rides. It's an anchor, an internal anchor. So now, if we've got intermediate filaments running from that desmosome to this desmosome, can this cell stretch in this direction? Probably not. And if they're going in all different directions, what are those intermediate filaments going to do? Stabilize the shape of that cell. Okay? So, it's almost more like a skeleton than any of the other parts. They're the most consistent, the most permanent, and they give the cell its shape. So they are tension-bearing filaments. They resist stretching. Yeah?
What are they made of?
Different types of proteins. That's why I said they're the most diverse. So to show a picture of them, there isn't one representative type, but it would probably be a lot like the intermediate filament or the microfilaments. It would be a lot like that, only different kinds of proteins and different sizes. Now, microscopic image. Here we have it. Here we have the microvilli. The core of the microvilli are microfilaments, in this case actin, and down here are the intermediate filaments. Pretty much everything else has been removed. And by the way, there are some cool pictures in your book where they actually label these structures with fluorescing dyes and then take pictures just of them. And it almost looks like spider web. They're that dense. I mean, like old spider web that's kind of piled up on top of each other. Anyway, any questions about this? Yes?
So microvilli, because they have microfilaments in them, does that mean they can just shorten to
In
get
this
flat
case,
again?
they're actually more stable. Like I said, not all of the microfilaments shorten and lengthen. Like in your muscle cells, they're called the thin filaments, or actin. They don't get shorter. They're pretty permanent.
Okay.
So sometimes they're more variable than others, and depends on this type of cell.
Okay.
So these would actually like
Similar, yeah, but inside the cell. All right, now the next thing I want to talk about is kind of a little strange because we're talking about cells, but now we're going to say, you know what? The next thing we're going to talk about is not inside the cells, it's outside the cells. But the reason why we talk about it is because this stuff is made by the cells. It's a product. It'd be like talking about wool on a sheep. Wool is grown by the sheep. You can cut the wool off. It's still a product of the sheep. All right? So, we're going to talk about cell walls. We're going to talk about extracellular matrix. And we're going to talk about cellular junctions, or what cells have to attach to other cells. All right? And then we will wrap it up and move on to the membrane. Now, first of all, cell wall is a generic term. Different types of cells have different types of cell walls. The one we are most familiar with is in plants, right? And that is made of cellulose. There are other things like chitin. Okay, that's another structural carbohydrate. Plant cells have cellulose. Fungi have different. Bacteria have cell walls. In fact, some bacteria are categorized by the structure of their cell walls. Gram-positive bacteria have a really thick cell wall, like 30 layers thick. Gram-negatives have just one layer of peptidoglycan. So if you take micro, you actually will look at gram positives and gram negatives, and you'll have to know the difference in their cell walls. So the contents differ from one type of animal or one type of organism to another. But plants, we can kind of say it's just basically cellulose. And fibers of cellulose, which creates these walls, which is very solid. But, let me just give you a little artwork here. Because these are solid, plant cells tend to be rather geometric. So you often see these cells being rather, relatively rectangular. And inside, that's the cell wall. And then just inside the cell wall will be the cell membrane. Okay? So that's right here. Now, when you look at it under a microscope, it would be continuous. Right? You wouldn't see any space in there. But I'm just putting that so you remember. Then, over here will be another cell. With another cell wall. And inside of that is another cell membrane. Okay, so far so good. Between each cell is going to be this middle lamella. And that middle lamella is a substance called pectin. Does that sound familiar to anybody? Pectin? Yeah, it's in jelly. That's what some people use to make jelly, thicken. In microbiology, we use the same type of substance, only instead of getting it from plants, we get it from algae. It's called agarose. It's a little bit easier to work with. But anyway, that middle, so this is quite a complex structure then. Okay? So we've got cell, membrane, wall, middle lamella, wall, membrane, cell. All right, so far so good. And as I said, a lot of times you use the structure of the cell wall for identification purposes, either chemically or structurally, but they also give a lot of stability to the cells. I mean, think about some of the biggest trees. They don't have bones. The only structure that those plants have is what comes from their cell walls. Think of like the sequoias, the redwoods, hundreds of feet high. And it's only because they have wood. And that, what is the wood? Wood is just an area that has a lot of cellulose in it, a lot of cell wall. Okay? So, the next thing, and this is kind of a general category of things. Extracellular. Extra means outside, right? Like they always talk about spacewalks as an extracellular movement or something, or extra capsular movement or something, moving outside. So the extracellular matrix would be something like if these were cells, it would be out here. In fact, some bacteria would look just like that. It's called a capsule. Other things, like our bones, the extracellular matrix is the hard part of our bones. The matrix of cartilage is what we think of as cartilage, that rubbery solid material. The extracellular matrix of tendons and ligaments is basically collagen fibers. Okay? The extracellular matrix of blood. Anyone know what the extracellular matrix is? Plasma. Now, that one is an exception to the rule because it's not solid. It's our only liquid tissue, but it follows the same guidelines. It has an extracellular stuff that's produced by the cells. Now, proteoglycans, things like collagen, and that's kind of a common substance, a common ingredient. Your skin has collagen in it, bone has collagen in it, cartilage has collagen in it, areolar connective tissue has collagen in it, dense regular, dense irregular. All these have collagen in it. It's a very common substance. In fact, about a quarter of all your proteins are collagen-based. But again, each substance is identified by variation in its matrix. Like I said, bone's matrix is different from cartilage, which is different from tendon, which is different from blood. In fact, some bacteria create what's called a slime, which is basically a substance they secrete that allows them to stick to surfaces. For instance, the bacteria that help create cavities stick to your teeth that way. They create a slime layer and then stick to it. Some, like this, create a very organized covering called a capsule. Helps them to avoid antibiotics. So again, the matrix is not a thing. The matrix is a number of different things that can be produced by cells that just happen to be exterior to them. Okay? Like I said with sheep, you might call their fur, their wool, sort of their matrix, if it was loose and just kind of floated around them. Okay? In fact, some organisms like bone cells actually paint themselves into a corner. They literally encase themselves in bone and they're stuck there. All right, so far so good? Now, the last thing I want to talk about, oh, by the way, when you take anatomy and physiology, we have a whole section on histology. You will go into the different kinds of extracellular matrices that are available. There's all kinds. So you just have to remember that it's outside the cells, but it's produced by the cells. Now, except for things like single-celled organisms like paramecium or euglena or amoebas or bacteria, cells need to work together for the most part. Even an egg cell has a multicellular covering on it. And there's a hypothesis that some of the sperm cells are working in teams as well. It's funny. You get a head start on some of them and the other ones start to swim in a circle to block the way for all the other cells that are coming up behind. So there might be some teamwork going on. But anyway, in order for cells to work together, they need connections. They need ways to connect to other cells. And these are 4 of the types that we're going to talk about. One is specifically found in plants. The other 3 are specifically found in animals. But this one and this one are very similar. The other 2 are more about connections.
Which two are
Plasmodesmata and gap junctions have almost the same function. All right, so let's start with Plasmodesmata. I don't know, the name doesn't really do much for me because it doesn't really tell you what it's doing. What we're looking at here is, if you look over there, it's some cell wall, middle lamella, cell wall, cell. Just like the picture I drew. But you notice there are connections that pass through that cell wall. So, now I enlarged it, but that is a plasmodesmata. It's a connection between the cytoplasm of one cell and the cytoplasm of the next cell. Really what we've just done by adding a plasmodesmata is making this into one giant cell. Because if the molecules are small enough to make it through that connection, they can pass unimpeded, unopposed from one cell to the other. Okay, it could be chemical signals, it could be nutrients, it could be water. But as long as you can make it through that plasmodesmata, this cell and this cell can communicate. Otherwise, what would happen? You'd have to diffuse from the cytoplasm, through the membrane, through the cell wall, through the middle lamella, through the cell wall, through the membrane, and into the cell again. Now just right through that tunnel. It's like a shortcut. It's almost like, a tunnel. Okay? That is a plasmodesmata. It's a way to link 2 plant cells next to each other. Yeah?
I'm going to kind of go back a ways.
Okay.
This diagram right here with the cell splitting, just down a little bit. Yep, just that right there. How does that cell ever split? I mean, how does it ever have 2 nucleus? That.
Well, bacteria would divide. This I was combining metaphors here, but the only reason I said this is because a lot of times it's even worse. In most capsules, there are 4 cells in there. But if you're talking about bacteria, they simply divide and start secreting their own coverings. If this was an animal cell, they wouldn't have a capsule. So I was just drawing that as an example. But what would happen is the cell would divide again, and then start to produce the capsule. It's defensive. All right, now, so plasmodesmata. Now that's in plants, and since plants held themselves together with the cell wall and then they communicate through the plasmodesmata, animal connections have to be a little bit different. Okay, but what I want to show you, and again, like I said with the original picture, you're going to find variation, and in these pictures they simply throw everything into them. You may not find two cells that have all three types of connections, but what we have here is a tight junction. These are the desmosomes, and these are gap junctions. Now I'm going to jump ahead a little bit, so if you got them, if you got the notes, I don't see a lot of people writing on notes. That's good. Good. Great. Anyway, so let's jump ahead to gap junctions. What is a gap junction? As the name implies, it's a gap from one cell to the other. It's a tunnel. Just like a plasmodesmata in a plant. So, what are they? It's a series of, see if I can recycle my picture here. It's a series of like 5 or 6 proteins that form kind of a cylinder. Okay? And if you look in your book or look at this picture, each of these is a tube that runs from one cell to the next. And through the middle of each of these tubes is a cytoplasmic connection. Again, just like plasmodesmata. So that stuff doesn't have to diffuse membrane space, membrane into the next cell. They simply diffuse through those cytoplasmic connections in a gap junction. They allow for molecules to pass rapidly. They sometimes allow for electrical signals to pass rapidly. So that is a way of allowing cells to communicate very quickly, more so than if they didn't have them present. Okay? So again, function of these two are almost identical. Now, structural connections, the two at the top then, are significantly different. Now, let's start with tight junctions. Anybody ever have a metal shop? You little spot welds where you put a spot weld and another spot weld. Tight junctions are a lot like spot welds. However, the difference is this. Let's say we're looking at the side of the membrane. The cell membrane, right? Outer cell, look. And that membrane is going to look like this, and it's going to have a protein wedged into it. It's called an integral membrane protein. It's within the membrane itself. If we take another cell next to it, like this, and it has an integral protein in the same location, and those two proteins stick together, the cell will literally fuse together at that particular point. Now, imagine if, let's say, we're inside of a cell, and we have a ring of proteins all the way around that's in the membrane itself. That means that everywhere around the outside of the cell, we can form a tight junction with the cell next to us. Doesn't that sound sort of like Tupperware? Right? It's got a seal on it. And again, like let's say you're going to glue something, right? Let's say you're gonna glue this, and you put a strip of glue right across there and slap it up on the wall and drop everything else on the floor. Do you see what I'm saying? As long as you've got that strip across here, you're completing a seal all the way across there. So as the two cells come together, like this, can anything get between them? No. If those cells are pulled apart, that stays a seal. That's why we call them tight junctions. They're not extremely strong, but they are complete. Okay? They form usually, now there are some exceptions, but for the most part, they form a continuous and impermeable barrier. Now, where in the body might we want something like that?
Around intestines and such, or things like
That's one of the best areas. Imagine this. Your stomach is making gastric juice at a pH less than 2. That's extremely acidic. That can eat through chunks of metal. Imagine if that leaked out between your cells and started just sort of fooling around with your connective tissue of your abdominal cavity. It would be nasty. It would be dangerous. Your stomach cells have tight junctions in them so that the stomach is a sealed container. So that all that gastric juice stays in the stomach where it's supposed to and doesn't leak out. So yeah, that's a good place for tight junctions. Okay? So anywhere where you want to produce a seal or a complete barrier, then you look for tight junctions. The last type of connection is called a desmosome. And I left this up here because I know I was going to come back to it. If looked at on edge, they look a lot like this, okay? But instead, you have membrane here and membrane here. The desmosome is on the inside surface, desmosome on the inside surface, and they connect by interdigitating proteins. But that looks pretty strong, doesn't it? And it is. But is it broad? No, it's actually very local. So again, let me just jump ahead here 1 second and show you a picture. Wait a minute. Oh, jump back. There it is. So here's one desmosome, and you can see how they're connected to the next cell. Here's another desmosome, here's a third desmosome, and you can see how they're interconnected. Those will rarely be broken. But, can you see what kind of problem there might be? What if you were to take those two cells, the one we're seeing and the one next to it, and pull them apart? What would happen? Not necessarily.
The desmosomes would stay intact if you tear the wall around it. Maybe misshape the wall.
That's what would happen. Imagine if you took two balloons and you put a spot of glue on them, and you stuck them together. And as you pull the balloons apart, what would happen? They would stretch, and they would stick only where the glue was. That's what happens to cells. As you pull the cells apart, they stay connected at the desmosome, but the rest comes apart. That means it's a very strong connection, but it's not a complete seal. Okay, it's a little stronger than a tight junction, but it doesn't act as a tight seal. All right? So you see how again, this would be used where you want stuff to stay together, but there isn't the necessity of having it complete. Your skin cells are held together this way. Muscles of the heart, cells of the heart are held together this way. Okay? In fact, the connection between muscle cells in your heart, cardiac muscles, is very cool. It has a lot of these things all combined. First of all, the surface between the two cells looks like this. We call them interdigitating folds. Or just like this. And when you see folds, what's the first thing you think of? Why would you need folds?
Surface area.
Surface area. So now you've got a lot more surface area for these cells to attach to each other. There are thin filaments that overlap from cell to cell. That's probably the strongest attachment. We talked about thin filaments before. There are desmosomes holding them together for a tight junction, or not a tight junction, but a strong connection. And on top of it all, we've got gap junctions as well. So think about that. These cells are held together very tightly, yet still have a lot of communication ability so that signals can get passed from one cell to the next. And by doing so, what happens? All of the cells that we want to respond usually respond at about the same time, which is why your heart can beat very regularly. So one part of the heart beats first, then another, then they just keep repeating it over. Your skeletal muscles don't behave that way. In fact, certain parts of your muscles will start before the rest. Okay? So, any question on that?
Membranes within the body, do they all have like a big junction, like a surface, like the pericardial sac or
Okay.
peritoneal membrane?
Okay, once again, when you talk about those membranes, now you're talking about different types of tissues. Membranes themselves will have connective tissue underneath and epithelial on top. Like a serous membrane, like the pericardial, is one thin layer of epithelial and connective. So that's mostly the fibers. It's really not cell-to-cell connection. It's matrix that's holding those together.
Okay. This is just on a cellular level.
Right.
Moderate. I guess I was going from cell to cell.
It can, it would.